Hello, my name is Professor Matthew Schmidt, and I'd like to welcome you to Genetics. In this session, a lot of things are going to come to fruition, or at least I certainly hope they will. We're finally to the point where we're going to see at the molecular level how translation occurs mechanistically. We've already got the concept down, and that's why I put the central dogma here, because we know that DNA information gets copied into RNA information, which we've already covered the mechanism of that. Then we've talked about the idea that RNA information is going to be copied into protein information. We've even discussed the code, the way that it must sort of happen. But we did not discuss the specific way the machinery operates, and that's what we're going to talk about today. And I really believe, I know personally, when I understood this, I felt it to be very, very powerful, and a lot of things really come together at this point, from original Mendel type experiments to everything basically. So we're going to look at the mechanism of translation. This is all part of the larger idea of how genotype turns into phenotype, if you want to think of it that way. We know that the expression of the information in a gene leads to a protein, and now we're going to see how that happens. We're going to use bacteria as an illustration for a couple of reasons. I've probably said it several times already, but all of this was actually discovered using E. coli bacteria. We know a lot about it in bacteria. We've subsequently learned quite a lot about it in other eukaryotes. So we'll do it the bacterial way, and then towards the end of this session, I'll talk to you about some general differences in eukaryotes. Okay? I don't want to keep you waiting because you've been waiting for this a long time. Now, in a certain sense, but don't make it too much of a sense in your mind. In a certain sense, the idea is similar to transcription, but only, in the following way. With transcription, we had a promoter which said start here. We had elongation of the messenger RNA, or whichever RNA. And then we had a terminator that said stop here, right? Well, it only makes sense. In protein synthesis, we're also going to have a start, an elongation, a continuing, and a stop, right? So, but the actual machinery is quite different for these things, all right? So remember what happened so far. A messenger RNA that's ready to be translated is going to be ready to go. It's going to be interacting with the ribosome. We don't have to go, we already just went over how that messenger RNA came into being. So let's look at the initiation step. How does it begin this process of translation? So first thing that happens, the messenger RNA binds to the small subunit of the ribosome, and this is a good point. A lot of people have the idea that the ribosome is sort of just pre-made with the big subunit and the small subunit, but this is not the case. The two subunits are not affiliated with one another until initiation occurs. Now, this has to be, just like transcription, we said it has to start right at number 1. We have to start at the exact right place here. So the way that gets done in bacteria is there is a complementary base pairing between the 16S ribosomal RNA and a sequence that is upstream of the AUG. Remember, AUG means start codon. And it's often referred to according to the names of its discoverers, the Shine-Dalgarno sequence. Often it's also just referred to as the ribosome binding site. But the point is, it's a part of the messenger RNA that is complementary to the ribosome. So when they base pair, it positions the AUG start codon very perfectly right in the ribosome, exactly where it needs to be. All right? So you can think the messenger RNA just, and I'll show you a picture in a moment, just came over and bound in the correct way to the small subunit. And then a tRNA, a transfer RNA, which has formylmethionine. I'm pretty sure we mentioned this, that so-called F-met. It's just a modified methionine in bacteria. That's the methionine that comes over. And an anticodon on that transfer RNA has to be like this. 3' UAC 5'. And the reason is, I think you get it, but if the start codon is AUG this way on the messenger RNA, right, that tRNA is going to come over, and we're going to need it to be complementary, so it's going to have to be UAC, but the polarities are the opposite way. So because that messenger RNA codon of AUG is there, always a transfer RNA with this anticodon is going to come over. In fact, that's why it comes over, because of the attraction, if you will, of the complementary base pairing. So now when we've got the messenger RNA, the small subunit, and then the tRNA all together, that's when the large ribosomal subunit joins the complex. And now we are ready for translation. Now, the ribosome is a fairly complex macromolecular structure. It's got about 43, I say about because there are definitely 3 big RNA pieces. There are about 40 proteins. I really don't remember exactly how many. But it has 2 functional sites, sometimes 3, but we're going to focus on the so-called P site. This stands for peptidyl, but we'll talk about why a little bit later on. Now, in the P site, that FMET tRNA that just came over is there. And then there's an A site. That stands for aminoacyl. It may not be obvious right away why this is the case, but we'll get to the bottom of it. Right now, it's empty. There's nothing in there. If your textbook or your professor speaks of an E site, the E site is something that does exist, but I think it complicates the analysis, so I'm not going to speak of the E site right now. It's really not necessary to understanding the process. Now, you might imagine this gets rather technical potentially, so I'm not going to speak about it a lot, but there are proteins called initiation factors, or IFs, that are necessary to facilitate these steps. Think of them as catalysts. They're not technically enzymes, but they're needed for this stable so-called initiation complex to form. All right? So let's take a pictorial look at what's going on here. They say a picture's worth 1,000 words at least. So if you want, we can just read this little description. It should be what we just said. The messenger RNA binds to a small ribosomal subunit near the initiation site, right? So it's not labeled here, but somewhere up here is the Schindel-Garneau site. See, when we make diagrams of this, it has to be a little bit off scale so that we can get the idea. And the reason I just realized that is if it's up here, it's not binding to anything, right? But we're making the bases look a lot bigger than they really are just so we can get the idea. So my point is the Schindel-Garneau sequence is somewhere upstream of the AUG start codon. The messenger RNA has bound to the small subunit so that the AUG start codon is in a very definite spot. Then a transfer RNA, again, just because AUG is there, a transfer RNA that has UAC as its anticodon is going to come over, and that is going to be carrying methionine, in this case, formal methionine, with it. So this is the beginning of the initiation complex. And this first tRNA is often referred to, as you might expect, as the initiator tRNA. So we have those together, and then we said the large subunit comes on over to form a functional ribosome. The ribosome is often referred to as the workbench on which a protein is made, and I think it's a very apropos sort of an idea. So once the large subunit comes over, a functional ribosome is formed. It's important for us to note that the A site and the P site, we're going to keep track of them as we go along here. So they're labeled here. This is the P site, and we said that the P site is the place where the first RNA that's come over exists, and that the A site is now empty. But you may notice, presupposing what's going to happen next, that now also, guys, I think you'll understand this. Obviously, there isn't a big space.